The process of protein synthesis has three steps. The first step is transcription, the second step is mRNA processing, and the third step is translation. So starting off with transcription,
DNA transcription is a process that involves the transcribing of genetic information from DNA to RNA. The transcribed DNA message is used to produce proteins. DNA is housed within the nucleus of our cells. It controls cellular activity by coding for the production of enzymes and proteins. DNA consists of four nucleotide bases: adenine, guanine, cytosine, and thymine that are paired together A to T and C to G to give DNA its double helical shape. DNA is transcribed by an enzyme called the RNA polymerase. Specific nucleotide sequences tell RNA polymerase to where to begin and where to end. RNA polymerase attaches to DNA at a specific region called the promoter region. In elongation, certain proteins called transcription factors unwind the DNA strand and allow the RNA polymerase to transcribe only a single strand of DNA into a single-stranded RNA polymer called messenger RNA. The strand that serves as the template is called the antisense strand. The strand that is not transcribed is called the sense strand. Okay. This is the poly A tail, which consists of three adenines that gives the mRNA its stability. Like DNA, RNA is composed of nucleotide bases. RNA, however, contains the nucleotides adenine, guanine, cytosine, and uracil. When RNase polymerase describes the DNA, guanine pairs with cytosine and adenine pairs with uracil. RNA polymerase moves along the DNA until it reaches a terminator sequence. At that point, RNA polymerase releases the mRNA polymer and detaches from the DNA.
Eukaryotic pre-mRNAs typically include introns. Introns are removed by the RNA processing in which the intron is looped out and cut away from the exons by NERPs, and the exons are spliced together to produce the translatable mRNA. The steps to pre-mRNA splicing are as follows. The intron loops out of the SNRPs and it goes in between the big ribosome subunit and the small ribosome subunit, which ultimately attaches the mRNA to the small ribosome subunit, which later attaches
the third step of protein synthesis is translation. Translation is when the information of mRNA is used to direct the synthesis of a polypeptide. A polypeptide could be assembled once the mRNA binds to the ribosome. The ribosome has the large subunit and the small subunit. The large subunit has two binding sites, the P site and the A site. tRNA carries an amino acid which will be incorporated to the polypeptide. Its anticodon is a triplet complementary to the codon on mRNA. So first, the ribosome must select the correct amino acid which it is specified by mRNA. The ribosome small subunit assembles on start codon for AUG. tRNA with the matching anticodon binds to the mRNA codon. tRNA now it's on the P site. The A site is ready for the second tRNA complementary with the second mRNA. Amino acid number 1 binds to amino acid number 2. The ribosome translocates the RNA in the A site to the P site, as shown. P site moved to the E site, where it is released. The A site is now available for the third mRNA to bind.
Elongation, and termination. In the first step, the small
The release-
attaches to the mRNA. The initiator tRNA binds the start, codon amino, binds the start, codon, and then thymine. Codon is the three bases of codon amino.
Translocation happens again.
tRNA has enzyme
So the E site.
complementary to codon.
Now, the release factor, which matches
After the start,
the codon and mRNA,
ribosomal subunit.
binds to the A site. The release factor carries no amino acid but facilitates the release of a polypeptide and mRNA from ribosome. Now translation is terminated. This is the polypeptide. And then now translation is terminated.